Welcome to the Endocrinology series and myself Dr. Amrita Bakshi from Ramjas College, University of Delhi. It is the second part of the Endocrinology series wherein we are going to understand the concepts of hormone receptor and mechanism of action. I'm sure you would be remembering what we have studied in the first part. We have understood about the discovery of hormones, who has to be credited for the discovery of the hormones, what are hormones, what are the classes of hormones. And today we are going to understand about their mechanism of action through their specific receptors. So in general, there are certain properties of the hormone receptors. Like most of the hormone receptors are proteinaceous in nature. Some of the protein receptors would be containing secondary modifications of carbohydrates. The receptors can get phosphorylated, which would in turn add to their molecular weight. Some of the receptors may form oligomers, even dimers, or some of them might not form any of the, may work as monomer. So in general, there are two components of hormone receptors, as you can see on the screen. One is ligand binding domain, which, as the name suggests, bind to the ligand or to the hormone. And upon binding of the hormone with its receptor, that leads to some conformational change. And then there is another component of hormone receptor which is called as the effector domain, which will then generate the biological response. As you can see in this figure, a hormone is going to bind to the ligand binding domain of the receptor. Upon binding of the hormone with the receptor, there is conformational change in the effector domain, and this effector domain is now capable of generating the biological response. So, there are two components of hormone receptors. One is ligand binding domain and the other one is the effector domain. So, now let us have a look at the types of hormone receptors. So, on the basis of their cellular localization, there are two types of receptors for the hormones. One is the membrane receptor and the other one is the intracellular receptor. The membrane receptor in turn could be classified into two parts for the ease of understanding. So there are some of the membrane receptors which gets internalized into the cell, but some of the receptors might not get internalized. So these type of membrane receptors which are not getting internalized, they could be in turn of two types: single membrane spanning receptors or multi-membrane spanning receptors. So membrane receptors you will find out for the hormones like luteinizing hormone LH, follicle stimulating hormone FSH, the receptor for insulin, the receptor for growth hormone, for the oxytocin, for antidiuretic hormone and the catecholamines. So the receptor of these hormones is found on the cell membrane. So now let us understand that what are the hormones which have intracellular receptors. So there are steroid hormones and thyroid hormone receptors which are found inside the cell, in the cytosol or in the nucleus. So the steroid hormone receptors would be for the glucocorticoid receptor. Glucocorticoid, an example of glucocorticoid would be cortisol. Then mineralocorticoid receptors, androgen receptors, estrogen receptors, progesterone receptors, T3 and T4 receptors, all of them have intracellular receptors. So let us now talk about the transmembrane receptors. These transmembrane receptors about which we have just studied, these are in turn of several types. The largest family of hormone receptor is constituted by the GPCR, which is G protein coupled receptor. So the receptors of GPCR includes FSHR, LHR, oxytocin receptor. There is another class which is of transmembrane receptors, and that is called as receptor tyrosine kinases. So the receptors for growth factors, for example, you might have heard about epidermal growth factors. Then there are PDGF, that is platelet derived growth factors. Insulin-like growth factors. The receptor of these hormones are basically tyrosine kinase associated receptors, and hence categorized into receptor tyrosine kinases. Then there are cytokine receptor family. So the receptors for growth hormone, prolactin, erythropoietin, leptin, the receptor for all these hormones belong to cytokine receptor family. The receptor for TGF-beta family receptor belongs to serine threonine kinase receptors. And lastly, for the transmembrane receptors, there are receptors for nitric oxide and ANP, which is called as guanylyl cyclase receptors. So, in general, now let us see that what a structure of a hormone receptor looks like. General structure of these transmembrane receptors include three domains. The first one is extracellular domain, which the name is suggesting would be lying outside the cell. It would be exposed on the outer surface. Then there is a transmembrane domain which would be present in the lipid bilayer of the cell membrane. And there is the third domain which would face towards the cytosol, and hence named as cytosolic domain, also referred to as intracellular domain. Now, in the extracellular domain, you will see that this domain has to basically interact with the hormone. It has to bind with the hormone. So, therefore, the extracellular domain will possess very high affinity binding sites for the specific hormones. Whereas transmembrane domain might be ranging from 1 to 7 hydrophobic domains, because it has to stay within the lipid bilayer. So therefore it would be made up of hydrophobic domains. And then there would be the cytosolic domain, which remains linked with the signaling proteins, which has to elicit the cellular responses so that some physiological response could be generated. So within this extracellular domain, there is one part which is the hormone binding domain, and there is another part called as hinge domain. Now let us understand their function. Hormone binding domain, as the name suggests, it would basically bind with the ligand or the hormone. It generally is present towards the N-terminus, and its role is, as I have just mentioned, that it will firmly and strongly bind with the ligand. Now, then what is the function of this hinge domain? This will lie after the hormone binding domain, that means towards the membrane, and it is involved in determining the specificity of the signal. With regard to the cytosolic domain, there are certain signaling domains. So there are several types of signaling domains which is not covered in this part of the lecture. So let us take an example of a follicle stimulating hormone receptor. So FSH receptor basically is a glycoprotein hormone receptor. It is a GPCR. You can see that there are several, by these orange drums, you can see these orange cylinders, these are actually the transmembrane domains. So there are these transmembrane domain ranging from 1, 2, 3 and up to 7. So there are 7 transmembrane domains. On the outer side, you can see that there is this ECD, which means extracellular domain, which includes hormone binding domain and the hinge domain. So there are several LRRs, which are leucine rich repeats arranged in this sequence ranging from 1 to 12, followed by an alpha helix shown with this drum you can see here. So this alpha helix is a component of hinge domain, while some of the initial LRRs along with these form the part of hormone binding domain. So as I have just mentioned, that you can see here that these green color are basically the hormone. So hormone is binding with the hormone binding domain, while this hinge region, which includes this alpha helix, is required for the specificity. After that, there is this transmembrane domain followed by the intracellular or the cytosolic domain. So we have seen about the general structure of these transmembrane receptors. Now what about the nuclear receptors? So now let's have a look at that. So the general structure of nuclear receptors includes three domains: the N-terminal domain, the middle DNA binding domain, and the C-terminal, that is carboxy-terminal ligand binding domain. Here a representative picture you can see for your reference, wherein an N-terminal domain followed by a DNA binding domain and a ligand binding domain is shown. But you must be wondering, what are these small H domain? What is this AF1? So now let us understand about each and every domain in detail. So there is this N-terminal domain, which is present towards the N-terminus and is usually variable. So for estradiol receptor, for androgen receptor, this NTD, that is N-terminal domain, is quite variable, and it contains this AF1, which is the trans activation function 1. Now what is the function of this AF1? It is generally involved in recruiting the co-regulators and the transcriptional machinery. So this is the function of AF1. In general, this NTD will modulate the cell and promoter specificity of the target gene activation. After this N-terminal domain comes the DNA binding domain. You will get to know about the function of this domain shortly in today's class only, wherein you will see that these type of receptors actually go and bind to the DNA. So there is a particular region of these receptors which will interact with the DNA. It is observed that this domain is quite conserved and is made up of some zinc finger motifs which are involved in recognizing and targeting the receptor to the specific DNA sequence. And that particular DNA sequence to which this hormone receptor would interact is called as hormone response element, HRE. So HRE is basically the sequence of the DNA to which this nuclear receptor will bind. Then there is a small hinge region which is shown with this orange color. You can see that it's a small region, it is variable, and it basically contains nuclear localization signal, this sequence NLS, and it plays a role in intracellular trafficking and subcellular distribution due to the presence of this NLS in this. With regard to the ligand binding domain, this region is again highly conserved and it has a sequence which encodes and attracts the association of heat shock proteins. It helps in dimerization and transcriptional activation or repression also. And as you can see here that this region contains the second activation function domain in this portion, which will assist in the robust transcription by interacting with co-regulators along with the AF1. Lastly, there is a C-terminal domain which is variable, and this variable C-terminal domain contains some retention signal for the sorting of the proteins. So now, by now you have understood that what are hormones and what are the hormone receptors. Now you have an idea that both hormones as well as hormone receptors belong to various classes. Now we must generate a generalized idea about how these hormones would act. So now let us move on to understand the mechanism of hormone action. And for this, let us have a general view. Whenever a hormone will come, it will interact with the receptor. And upon binding of the hormone with the receptor, there would be conformational change in the three domains. This conformational change is called as the signal. Now this signal will get transduced and activate the signaling molecules. And these signaling molecules will then act on the effector proteins, which will finally modify the cellular function. So together, from the generation of the signal to transduction of the signal to the effector proteins, this is what is referred to as signal transduction pathway, while the last part wherein the changes in the cellular function would take place is a part of cellular response. So for understanding the mechanism of action of the hormones, we will classify and use the same classification as we have studied for the hormones, that is the water soluble and the lipid soluble hormones. So you can understand that the plasma membrane of the cell is made up of lipids. So it would be definitely very easy for lipid hormones to cross that lipid bilayer. But the situation is not so easy for the water soluble hormones. So let us first understand about the mechanism of action of the water soluble hormones, which includes our amines, peptides, proteins and eicosanoids. So in this figure, what you can see here, that from the blood capillary the hormone is traveling, and now there is a cell which is a target cell which is expressing a specific receptor for this hormone. Now as soon as the hormone will bind to its receptor, there would be the proteins present on the inner side of the cell. So there are G proteins which will get activated, which in turn would stimulate the adenylate cyclase. Now, as the name is suggesting, that this is an enzyme which will convert the ATP into the cyclic structure, and that cyclic structure is none other than CAMP. So, now let me stop you here. The hormone first binds with the receptor. This hormone is actually the first messenger which will give the signal to bring some change in the cellular function. Thus, hormone acts as the first messenger or the primary messenger. When this CAMP is generated, this is the second messenger. And now the function of the hormone would be carried out by this CAMP molecule. This CAMP would in turn activate the protein kinases, and as the name suggests, kinases will always add a phosphate group to the proteins. So these kinases will take up phosphate from the ATP and will add that phosphate to the protein. Now this phosphorylation of the target protein might activate it or might inactivate it as well, thereby bringing modification in the cellular response. So this is a general scheme how the water soluble hormones act, wherein the first messenger is always the hormone while the second messenger is the CAMP. Now what is interesting here is that I have told you in the first part of the series that hormones always work in very small concentrations, micromolars or even less than that. Now you must be wondering that how a hormone present in nanomolars, picomolars, micromolars could bring so many changes that we see inside our body. So actually there is an amplification of the signal which takes place at the level of signal transduction. So when the hormone binds with its receptor, it basically will, let us take an example, one hormone is binding with one receptor, but it will activate the adenylate cyclase, and this one adenylate cyclase will in turn activate 10 CAMP. It will generate 10 CAMP. Then these 10 CAMP molecules will in turn activate hundreds of PKs, that is protein kinases. These hundreds of protein kinases will again amplify the signal and will phosphorylate. So this is how the amplification of the signal takes place, and hormones remain effective even at very low concentrations. I hope this is clear that how water soluble hormones act. Now let's move forward to understand the mechanism of action of the lipid soluble steroid hormones or the thyroid hormones. So you can see here that in blood, these hormones which are lipid soluble, they definitely cannot just flow in the bloodstream like that because they are not water soluble. So they require a transport protein as shown here in this figure. So there is a transport protein which is helping this hormone in traveling through the bloodstream. As soon as this hormone reaches to its target cell, this lipid soluble hormone gets dissociated with its transport protein and enters the cell. Now inside this cell, you will see that the receptors are present either in the cytosol or in the nucleus. For example, in this figure, the receptors, these are the nuclear receptors which are shown here as monomers. As soon as the hormone comes and binds to the receptor, the dimerization of the receptor takes place. So now this is basically an activated receptor hormone complex. Now I hope you remember that there was DNA binding domain within the nuclear receptors. So these DNA binding domain will now interact with the DNA or the specific sequence called as hormone response element. And now upon binding with the DNA, there will be transcription followed by translation and production of the new proteins. So in this way, in general, the lipid soluble steroid hormones or thyroid hormones work. This mechanism of action wherein the hormones, the steroid hormones or thyroid hormones directly passes or crosses the cell membrane and binds to the intracellular receptor is what is called as the classical pathway or classical mode of action of the steroid hormones. But you should be aware of the other mode of action of steroid hormones, which is actually non-classical. So in one part here you can see the same mechanism of action which I have just explained you, wherein you can see the steroid is entering the cell. So this is the plasma lipid bilayer, the plasma membrane lipid bilayer. The hormone is entering, interacting with the SHR, that stands for steroid hormone receptor, and thereafter it is getting dimerized, binding to the hormone response element, and thereafter the transcription is taking place by recruiting the co-transcriptional activators and so on. So this is the classical mode of action. But what happens in non-classical mode of action, that these steroid hormone receptors are found to be present on the membrane also. So here you can see that M, small m, is here showing or representing membrane. So this is a membrane steroid hormone receptor. the hormone can bind to this receptor and will thereafter activate n number of secondary messenger molecules. So this is a non-classical mode of action, and these classical and non-classical mode of action are also called as genomic and non-genomic mode of actions. So in this lecture, what you have got to know is that what are the different types of hormone receptors? What is the general structure of these receptors? So we have seen that as we have classified the hormones as water soluble and lipid soluble, in general we are seeing that receptors can be studied as a transmembrane or nuclear receptors. We have seen that the transmembrane receptors have their extracellular, intracellular and transmembrane domains, whereas the steroid hormone receptors also have a number of domains including DNA binding domain, N-terminal domain, C-terminal domain. And after understanding the general structure of the hormone receptors, we have seen in today's lecture and studied about the mode of action of these hormones. So we have seen that there are two different pathways for water soluble and for lipid soluble hormones. In case of water soluble hormones, we have seen that there is a receptor on the membrane because the water soluble hormones cannot cross the lipid bilayer. Whereas for the steroid hormones or the lipid soluble hormones, there are two pathways, classical and non-classical mode of actions. So I hope this has given you an overview of the field of endocrinology, and from next series onwards we will focus on particular each and every endocrine gland. Thank you.